Shalvarajan sir? Please unmute.
Shall we start?
2 minutes are there.
Okay.
Wait for 2 minutes.
Okay.
Sharp at 9 we will start.
Yeah.
Co-chairman Dr. Dhananjay will be joining within few minutes.
Okay.
So you can start the program sharp at 9 o'clock.
Okay. Sangeeta has joined, I think, sir. Good morning, Sangeeta.
Very good morning, sir.
Good morning. Mahantesh Naik has joined. Good morning, Mahantesh.
Good morning, sir.
Good morning.
Dr. Sangeeta?
Yes, sir. A very good morning to all the delegates, speakers, and participants. I welcome you to the third day of this international e-conference, where we start the theme number 6, Omics and Informatics in Plant Pathology today. For this particular session, we have our chairperson, Dr. R. Selvarajan, sir. I will extend a warm welcome to you, sir. And I would like to introduce you to our chairperson, Dr. R. Selvarajan, who is working as a principal scientist in ICR NRC Banana. He's a renowned virologist who has done a lot of work on banana viruses. He has handled or completed about 11 projects, and he currently has 5 ongoing projects. He has worked on the complete genome sequencing of banana viruses, and he's got many awards to his credit. Few of them, to name a few of them, he's a Fellow of Horticulture Society of India, G.H. Sannidhi Award from IPS, and he is the editor of Indian Phytopathological Society and editor of Virus Diseases, and he has a total of 155 publications to his credit. He has guided about 3 PhD students and 74 MPhil and MSc students. A very warm welcome to you, sir. It's an honor to have you here. And we have our co-chairperson, Dr. Dhananjaya, who is a principal scientist in IHR Bangalore in the Department of Genetics and Plant Breeding, and who has released about 5 varieties of aster, who has about 23 years of his experience, and has handled about 5 to 6 projects worth 25 crores. And he has 75 publications to his credit, and he's a life member of about 5 societies. I welcome you also, sir. And I welcome the rapporteurs, Dr. Mahantesh Naik and Dr. Ambika. I also extend a warm welcome to the key speakers, Dr. Murli Raghavendra Rao and Dr. Professor Hanu and Papu and Dr. Narendra Kadu. Now, I hand over to Dr. Selvarajan, sir, to take over the session.
Yeah, thank you. Good morning. Thank you, Sangeeta. Good morning to all of you, and good evening for the keynote speakers joining from USA. It's a very, very interesting topic, and we are going to have 2.5 hours with you in this session. And in this session, we are going to have 3 keynote speakers, and each spanning around 30 minutes, and there will be another 5 oral presentations with us. And omics approach is very, very important, and without a bioinformatics support, this omics cannot be published. So you may have a lot of wet lab work, and without bioinformatics specialists joining. Without their support, nothing can happen. And omics approach, all of you know that, lot of developments has happened for the last 2 decades. Look at the transcriptomics, proteomics, and metagenomics, and high throughput sequencing has made a lot of developments in the plant pathogen interaction, and many papers. I think more than 10,000 papers you would have seen the last 2 decades, and these omics further has come out with a new omic approaches like ionomics, epigenomics, lipidomics, genomics. There are many omics keep on coming, and which gives a lot of understanding over plant defense mechanisms, and the understanding on the plant host interaction, and again the microbiome. And that's where another very important thing, wherein we all talk about biological control. We use biopesticides and its interactions with the host and the pathogen. And a wonderful knowledge has been generated for last 2 decades by worldwide scientists, our plant pathologists, plant virologists, and plant protection specialists, and joining with the biotechnologists and the statisticians and bioinformatics specialists. A lot of understanding has happened over the past 2 decades, and not taking too much time, as I said earlier, we can have 27 minutes talk and 3 minutes for keynote speakers, and for oral presentations, as usual, 10 minutes. And now I welcome the first keynote speaker, Dr. I welcome my the co-chairman and the rapporteurs for this session, and the key speaker, Dr. Murali Raghavendra Rao. I would like to just introduce before his talk. Dr. Murali Raghavendra Rao received a bachelor's degree in agriculture and a master's in horticulture from University of Agricultural Sciences, GKVK, Bengaluru, before earning his PhD in plants, soils, and insects from University of Tennessee, USA, with the emphasis in plant molecular biology. He is also received MS in statistics from the University of Tennessee, along with his PhD. So, after a short stint of University of Tennessee, a postdoctoral.
Sir Selvarajan, sir, you have muted, sir. Kindly unmute. Yes.
Oh, I am sorry.
Yes, sir.
I am very sorry. I think okay. It's totally, it was unmute, no? What I was talking.
No.
Okay,
Yes,
okay.
sir. Please.
Oh, I am very sorry.
No, it's okay, sir.
You would have just told me. Okay.
It's okay, sir.
Okay. I will make my what I talk just 1 minute. I will just tell me. So this omic approaches has really made the understanding and the plant. No. Did I whatever I talked was there? It was muted.
Yes, sir. It was all there. Only the last few sentences you were.
Okay. Thank you. Sorry. So it was muted by, I think, host. Sorry. Okay. So Dr. Raghavendra Rao, did I mean? Okay. He is one of these. As I said, that he joined in Purini Technical Innovation Division as a manager of statistical services. Since then, he has aided in the development of several successful products for Purina. He currently serves as a director of data intelligence. He and his team cater to statistical and data needs of over 30 PhD scientists at Purina. He is also an accredited professional statistician accredited by American Statistical Association. It's a very big thing. And I welcome Dr. Raghavendra Rao, and we are eager, and all of us are very eager to listen to you. Please, you can take over.
Okay, sir. Thank you, Dr. Selvarajan, sir. Can you guys see my screen?
No, yeah. It's starting to share content. Yes.
Okay. Are you are you seeing the slides, Sangeeta? You know which.
There's something your chat box is also seen today. Good morning. Yeah. Outline.
Okay, I need to change. Give me 1
No.
second.
Yeah.
Okay now?
Yes. Now it's okay.
Okay. Good morning, everyone. Before I begin, I would like to thank Dr. Devappa sir and Dr. Sangeeta for giving me this opportunity to speak. Today, I'll be talking about the role of bioinformatics in plant microbiome analysis, and also how we can leverage the cloud infrastructure to streamline the bioinformatics process. So I've broken down my presentation into 4 sections. I'll start off with a refresher on bioinformatics and its goals. Then I'll talk about plant microbiome and its importance and the role of bioinformatics in plant microbiome analysis. And then I will introduce cloud computing. And then I'll wrap up my presentation with a few examples on how we can use cloud to improve the bioinformatics process. So what is bioinformatics? Bioinformatics is an interdisciplinary field of science which borrows expertise or knowledge from computer science, mathematics, and statistics to acquire, store, analyze, and interpret biological data. So over the last few years, the field of bioinformatics has grown exponentially due to 2 main reasons. One is significant advancements in life sciences or biological research through great strides in data collection methods or technologies such as next generation sequencing. And secondly, due to significant advancements in computer science. We have made huge improvements in sharing, processing, and storing data, and also development in high performance computing. With such progress, you can imagine the numerous applications that bioinformatics might have within life sciences. So you need bioinformatics, like Dr. Salvarajan also mentioned, you would need bioinformatics for, making sense of all the large, huge amounts of data, sequence data that is generated from next generation sequencing, to understand gene and protein expression, to identify microbes and then study microbial diversity, and also to predict and analyze 3-dimensional structures of biological macromolecules, and also to understand the complex pathways, and last but not least, biodiversity and phylogenetics. Now let's look at the goals of bioinformatics. I mentioned that we are generating enormous amount of data. The question becomes, how can we make sure that we store this data, organize this data in such a way that researchers can access it and utilize and advance their research? One way is to deposit all that information into databases. And this slide shows some of the databases that bioinformatics uses. Some of the popular ones are GenBank from NCBI, which hosts nucleotide sequences from over 300,000 organisms. And then you have UniProt from the UniProt consortium, which comprises more than 500,000 manually curated entries. And then you have databases from premier institutes such as European Molecular Biology Laboratory or Swiss Institute of Bioinformatics. The second most important goal is to develop software and tools to utilize all that information, and also to make sense of all the data and we are generating. So this slide shows some of these tools. So we have tools for such as BLAST for sequence similarity search, Motif for sequence motif search, and you have TX search for querying a taxonomy database, and then you have ClustalW for multiple sequence alignment, Pictree for phylogeny, and then you have submission tools such as Bankit, Sequin, or Sakura, and some analysis tools such as ArtFinder and MSD4. Now we'll move on to plant microbiome. How do you define plant microbiome? Plant microbiome is referred to having an above ground section and a below ground section. So the above ground section is referred to as the phytosphere or phylobiome, that comprises of leaves, stem, and all the reproductive parts. And the below ground is referred to as rhizosphere or rhizobiome, which comprises roots and small amount of soil around the roots, maybe up to a centimeter. Then you also have bulk soil microbiome, which can extend up to 2 centimeters or more. Then you have epiphytes and endophytes. Epiphytes are the microbes that harbor the plant surface, and the endophytes are in the epidermis. They can be between the cells or within cells. Now, what are the factors that influence plant microbiota? So you have plant or host factors, which is species specific, and different crop cultivars can have distinct microbiomes. And then other factors that influence in this category, are age and developmental stage. And then you have microbe-microbe interactions, where microbes can interact with plant-associated microbes in several different ways. And might exhibit mutualism, parasitism, commensalism, and which can have significant impact on the plant microbiota. And then finally, you have environmental factors such as climate, soil, and cultivation practices, which can also impact plant microbiota. And important thing to note here is all these different categories of factors, are interlinked, and together they render the microbiome community structure rather unique. So now to the obvious question, why study plant microbiome? Plant microbiome research, has shown that the plant-associated microbes often increase the resource uptake and offer plants with novel nutritional and defense pathways, which can have direct impact on functional traits such as leaf area, leaf nutrient levels, and shoot-to-root ratio. Which is not confined to just a single plant, which can extend beyond and also reach complete ecosystems. Now, plant microbiome research discoveries can also fuel advances in sustainable agriculture, through development of biofertilizers, biocontrol agents, stress protection products, and probably help us reduce the use of chemicals. And it's also known that plant microbes have a positive impact on yield. So better understanding here might help us increase agriculture production, something to keep in mind for future breeding or biotechnology strategies. And yesterday we heard Dr. Dinesh Kumar mention how plant pathogens are a constant threat to global food production. They are responsible for 20 to 30% of global crop losses. So now recent research has shown that plant-associated microbes confer extended immune functions to the host, and better understanding here might help us prevent outbreak of plant disease. Now, you might have heard of probiotics, prebiotics, and symbiotics for humans. Now, there is research proposed to develop these products for plants. And then there is considerable interest in studying the interaction of plant microbiome with human microbiome, and can have a major impact on human health. And last but not least, there is scientists are also interested in studying microbes associated with medicinal plants. It's been recognized that a significant number of these phytotherapeutic compounds are either directly produced by these associated microbes or in interaction with them. So now we know that plant microbiome is important. Now let's look at the role of bioinformatics in plant microbiome research. So one of the research priorities in microbiome research is defining the core microbiome and the microbial metagenomes. So I mean, there's a lot going on this slide, but you can just focus on the two red boxes here. Those are the two approaches that are used in research. First one is a single gene target approach or amplicon sequencing approach.
Sir, sorry for disturbing. Sir, sorry for disturbing, Dr. Rao. I request all the people to mute except the speaker, and it is giving. Kindly see your mic. Please mute. All of you, please mute. Only the speaker mic should be on. Please, sir, you continue, sir. Sorry for disturbing.
No problem, sir. So there are two approaches in microbiome research. The first one is the single gene target approach, or amplicon sequencing, where you would sequence target genes such as 16S or 18S ribosomal RNA, or internal transcribed spacer regions, genes. So although fast and cheap, this approach is very specific and does not provide more information on all the other genes from the microbial genomes that remain unsequenced. Then the second approach is the whole genome shotgun metagenomics approach. Compared to the single gene target approach, this involves sequencing the whole genome of all the microorganisms in your sample. This approach is known to provide better phylogenetic resolution and also enables gene function prediction. So to wrap up this section, we need plant microbiome research, and bioinformatics is key for this research. And I want you to note that the amount of data that we handle and process makes this step very computer intensive and time consuming. So now let's look at cloud computing. So what is cloud computing, right? So it is nothing but delivery of computing services or resources over the internet. It could include servers, storage, databases, networking, software, analytics, and lately intelligence as well. I want to play a short video. Give me just one second.
Cloud computing is the on-demand delivery of IT resources via the internet, with pay-as-you-go pricing. Instead of buying, owning, and maintaining physical data centers and servers, you can access technology services such as computing power, storage, and databases on an as-needed basis from a cloud provider like Amazon Web Services. Organizations of every type, size, and industry are using the cloud for a wide variety of use cases, such as data backup, disaster recovery, email, virtual desktops, software development and testing, big data analytics, and customer-facing web applications. For example, healthcare companies are using the cloud to develop more personalized treatments for patients. Financial services companies are using the cloud to power real-time fraud detection and prevention. And video game makers are using the cloud to deliver online games to millions of players around the world. With cloud computing, your business can become more agile, reduce costs, instantly scale, and deploy globally in minutes. Cloud computing gives you instant access to a broad range of technologies so you can innovate faster and build nearly anything you can imagine. From infrastructure services such as compute, storage, and databases, to Internet of Things, machine learning, data analytics, and much more. You can deploy technology services in a matter of minutes and get from idea to implementation several orders of magnitude faster than before. This gives you the freedom to experiment and test new ideas to differentiate customer experiences and transform your business, such as adding machine learning and intelligence to your applications in order to personalize experiences for your customers and improve their engagement. You don't need to make large upfront investments in hardware and overpay for capacity you don't use. Instead, you can trade capital expense for variable expense and only pay for it as you consume it. With cloud computing, you access resources from the cloud in real time as they're needed. You can scale these resources up and down to grow or shrink capacity instantly as your business needs change. Cloud computing also makes it easy to expand to new regions and deploy globally in minutes. For example, Amazon Web Services has infrastructure all over the world, so you're able to deploy your application in multiple physical locations in just a few clicks. Putting applications in closer proximity to end users reduces latency and improves their experience. no matter your location, size, or industry, the cloud frees you from managing infrastructure and data centers so you can focus on what matters most to your business.
Okay, so back to the presentation. So now we learned, what is cloud computing. So now there are three types of cloud computing that these cloud providers offer. One is infrastructure as a service, where you would rent individual virtual machines or servers or databases. Then you have platform as a service, where you would rent the entire platform that you would use to develop, test, deliver, and manage your application. Then you have software as a service, where the cloud providers host and manage your software application, typically for a subscription on a subscription basis. Um, so which is very easy. And a good example for that is email service. Now let's look at the benefits of cloud computing, right? So cost effective because you don't have to buy and maintain any computers. You can scale up or down as you need it, and then your hardware and software is always up to date. You always get peak performance, and then there'll be increase in productivity because you don't have to spend time anymore to manage your infrastructure. Then reliable in terms of data backup and disaster recovery, and speed because you can turn on the resources whatever you need in a matter of minutes. And I'll save the security piece for the next slide. So whenever I mention cloud, the first concern people raise is, how safe is my data, right? How can I trust cloud? Now, this information that you see on the slide is from Microsoft Azure website. So now Microsoft spends more than 1 billion US dollars, which is more than 7,000 crore rupees in cloud security R&D. And they employ 3,500 cybersecurity experts who are making sure that your data is safe. And then they have very strict policies in place that protect your privacy. And then they provide over 90 compliance offerings. What I mean by that is, they meet or exceed standards set by the US government, global and industry specific certifications, including Government of India's Ministry of Electronics and Information Technology standards set by under the Meghraj Cloud Initiative. And not to mention, they analyze 8 trillion threat signals every day, which is like 1 lakh crore cloud security threat signals. And not to mention, there are more than 95% of Fortune 500 companies use Microsoft. Knowing all of that, would you trust cloud? I would. I would also say that my data is much safer on the cloud than a local computer. So this slide shows industry-specific cloud companies. This is not the latest information. I'm sure the 2020 landscape would look much busier, crowded with a lot more companies. And then these are the major cloud providers. Amazon leads the market, and followed by Microsoft and Google. So now let's move on to how we can use cloud for bioinformatics, right? I would like to spend a minute, reflecting on the drawbacks of using local computers for bioinformatics, especially microbiome analysis. So using local computers, you would run into prolonged runtimes, and your future analytics could be delayed. And when it comes to storage, given the amount of data this research produces, you might run into challenges, where you cannot efficiently store your data. And the major drawback is sharing with your collaborators. Uploading and downloading these huge microbiome data sets can take days, if not forever. And not to mention your overhead of computing infrastructure, performance issues and reproducibility and efficiency issues. Now, to overcome all those drawbacks, researchers have opened up and started to explore cloud for their bioinformatics needs. So these researchers, did a proof of concept study where they try to set up a bioinformatics pipeline on Amazon Web Services, and this is the outline they laid out. You would first, start with getting together an interdisciplinary team that would comprise researchers, data scientists, bioinformaticians, external collaborators, and probably people from your computer science department as well. Then all that microbiome data that's generated is incrementally uploaded onto the cloud. In this specific architecture, they had two components: storage component where you would securely and store your data so that you can access it better, and then the computation section where you would host all the bioinformatics software such as Chime, R, and any other packages that you would require for analyzing the bioinformatics. So this is another example. So these researchers developed a tool for open source tool for metagenomics profiling called Flint. So when I say metagenomics, as I mentioned earlier, you are sequencing the whole genome of thousands of microbes, and you can imagine the amount of data you. And the first step here is assembly and binning, and that step is very computer intensive and time. Now, to do that, instead of having one big computer, what these researchers did was they used Apache Spark cluster, which has built-in parallelism. They used a concept called parallel computing. Instead of one big computer doing all that work, they distributed the work among several different smaller computers. So this is not only cost-effective, but also saves a lot of time. And this is a smart way to do this. And this kind of architecture, can only be possible on the cloud. I cannot even think of setting this up locally. It could be a nightmare and very, very expensive. So this is another example that we at our company are planning to deploy on Amazon Web Services. This again, similar to the previous example, we are using distributed computing here to perform all the computer intensive steps. Especially here, we set this up for the data to study. So if you have 96 samples from 96 well plate, you would have one computer dedicated to each sample. You would have 96 nodes or 96 computers all performing the analysis at the same time. So that you save time and money, and after all that, everything is regrouped and presented back to the primary computer or node. And again, this kind of architecture is possible only on the cloud. So to recap, plant microbiome research generates a lot of data, and then bioinformatics is absolutely essential and key for this research. And given the amount of data that is handled and processed, cloud offers secure, reliable, and cost-effective model. That's what I had. I would like to thank Manohar Saad and Veena for helping me put this presentation together. And with that, I'll take any questions.
So, thank you, Dr. Raghavendra Rao, on very, very insightful talk. And he has completed.
You're on mute.
Please don't unmute me. Someone
Yes,
is
sir.
unmuting. Yeah, continuously. I don't know. Yeah, please. This is open for questions. Please, those who raise questions, it's a good opportunity and it's a wonderful talk by Dr. Murali Raghavendra Rao. And those who are interested immediately can ask questions, can raise.
Murli,
You can raise
hello,
your
sir.
hand, please. Yeah.
Murli, sir, congratulations for good talk, nice presentation, and good informative. It is database is a future science, no, sir. Yeah, how to manage the data, because the managing a data is a big task.
So that's why you need you can easily do that. You would need somebody with an IT background, somebody who knows computer science. Then you can easily rent what you need on the cloud and then upload your data over there. The beauty of cloud is you pay for whatever you use, and the storage is very cheap these days, and all the services are very competitive. You have a very healthy competition in the market, between Amazon, Microsoft, Google, IBM. So it's very cheap to do that. You just need the right expertise to
Thank you.
be able to do that. It is not future, it is happening now, and it can be done.
I think Nakiran, yes, I got a question.
Questions? No questions, please.
Okay. Any questions, please?
What's your PowerPoint? Your presentation.
Yeah. That's good. Any questions?
ಹೇಳ್ಬೇಕು sir.
Any questions? But it's very nice that the cloud computing, the security is very, very good. That's what Dr. Rao said. And this is really, everyone has that question whether what we keep in cloud, whether it will be looted by someone for there. So that's very interesting that he has cleared the doubt that it's very safe. And 7,000 crores has been being spent for security alone, the cloud computing. That's very interesting information. Um, any questions, please? It's a good time to talk questions. Students also can come forward and ask questions, or even chat box. I am not finding any questions in the chat box.
I would also like to mention that, sir, these cloud companies, cloud providers, they work with you, very nicely. So they entertain you, and then you can do a proof of concept study, just to figure out, what you need. And most of these companies do it for free to help you understand, what you need and what you don't need, how you can set up your need.
So it's a very nice and informative presentation.
I think the person who is asking question can tell about them, then ask questions. No? Okay, I think, thank you, Dr. Rao. We will go for a next talk. Thank you very much. And we'll be going for next talk. Yes. Yes, Dr. Sankarappa, please.
There is some problem with network.
Okay. But Dr. Pappu sir is, Professor Pappu is with us. There's a network problem with Professor Pappu could not.
Audio issue is there.
Good evening, Dr. Pappu. Good evening, Dr. Pappu. I think he could not hear our voice. Dr. Pappu, could you hear? Dr. Pappu, could you hear? I think he's sitting. he's setting his system. I think we can give a few minutes to him to join with us. Yeah, he's here.
Yes.
Voice.
Thank you, Murli.
Sangeeta.
Professor Devappa, there is some audio issue is there.
No, there is no problem from host side. The problem from other side only, because here everything is okay, no problem.
No, it's fine. Could you hear Dr. Prabhu? Good evening. Dr. Prabhu could not hear actually.
Yeah, that is the problem.
What to do? There is some serious audio issues there, network issues there.
Dr. Selvarajan, I have sent message to Dr. Pappu.
Yeah, I
I'm trying to call him
I have sent, but he's not replying to my message. I
Yeah,
think
he's
he's sitting right now actually.
He's joining.
Good evening, Dr. Prabhu. Yeah, I think he's now. Yeah. Good evening, Dr. Prabhu. Good evening. Dr. Prabhu, could you hear? Oh, your voice we could not hear. I think some mic problem. You can saturate your mic probably.
Chat
I in chat. I sent a message.
problem started right shot. Hello.
Dr. Prabhu, yeah, we could not.
Dr. Selvarajan, is there any response from Papu?
Oh, Dr. Prabhu, yeah, no. I am just sending a message continuously to him, but he could not be able to hear.
Just
Sir,
let us
Dr.
wait.
Pappu has muted, sir. He should unmute.
Yeah, and I was asking him to unmute also. Why don't you, can we do that unmuting? From who is the handling this muting, unmuting? Someone unmute, yeah. Papu,
Yes, sir.
good evening, Dr. Papu. I think he is listening us, but we could not listen to him. There's a mic problem. He is listening, yeah.
He's listening, yes, sir.
But we could not. There's a problem in mic, probably. Dr. Papu, kindly look at your mic. The mic.
Earphone, sir.
Earphone mic, I think. Maybe some problem in the mic only, yeah.
had similar problem during my DBT presentation. Maybe he should try not to use headphones.
Oh, that's
Yes, sir. Yes, sir.
maybe he has to use. If it is laptop, straight away he can use the laptop
He
mic
can
as
straight
well.
away talk.
Yeah, if it's a desktop only, this problem comes. But it's not seen now. No, Chili, you have his number, personal number. Can you?
Selvarajan sir,
Uh,
you may rejoin. He is disconnected.
Disconnected. Yeah, I think who is the host and again connect him. He's disconnected. So what we can do is like shall we have the next talk, then we can make him to join.
Yes,
Celia,
sir.
can you talk to Celia? Please talk to Papu and yeah, he can join after 20 minutes. Ask him to set it. Okay, let us go for next talk. Okay.
Yes.
Okay, that's good. Next talk is by Narendra Kadu. Whether Narendra Kadu is here? Dr. Narendra Kadu?
Sir?
Dr. Narendra Kadu?
Is there, sir?
Is there, no?
Yes, it is there, sir.
Okay, I think still he's setting the problem. I think Narendra Kadu can give his talk.
Papu sir back.
But Narendra is back?
It is showing back. Yes, he is back. Papu sir back.
Oh yeah, Papu sir is back. Good evening, Dr. Papu.
Can you hear me?
Good. Yeah. We could hear. Yes.
Okay.
Thank you. We could hear some technical glitches probably. I request all of you to mute except the keynote speaker, please, because there is a background noise is coming. Hello, when you are muting everyone, you are muting mine also. What?
Sir, Selvarajan sir, kindly unmute, sir. Yeah, done, sir.
Yeah, someone is muting. Unmuting my. Okay. Good morning and good evening to Dr. Hanu Papu. Professor Papu, I think it's okay, no? I think, Dr. Papu, could you hear me?
Yes.
Oh, thank you. I would like to tell you brief about Dr. Papu before his talk. And that all of you know, almost every plant virologist knows about Professor Hanu Papu. He is a IRI alumni, Professor Anupam Verma's student, and he used to be in all our conferences conducted by the Indian Biological Society, and he is one of the a well-known virologist, and he did his BSc Agriculture from Bapatla, then IRI for Masters, and he did his PhD in University of Alberta, Canada. If I look at his awards and awards, it would take another 20 minutes for me to tell his honors and other things. A few of his is a Professor B.P. Paul Distinguished Chair, Indian National Science Academy, and Fulbright Distinguished Chair, and he is a Fellow of National Academy of Agricultural Sciences. That in 2016 he bagged that, and he is a Alexander von Humboldt Research Award he received in 2018, and he is a Chuve Endowed Chair, and he's a Fellow of American Phytoweb Society. But he has got a very big awards list, and I don't want to read one after the other. But his contribution to the virology research is very, very great. And straight away, I invite Dr. Papu to give his talk on gaining insights into the plant virus interactions during the omics era. So straight away, I request Dr. Prabhu to share your presentation. Go ahead, please.
Good morning, everybody. I apologize for the technical difficulties. We routinely use Zoom here at WSU. So this WebEx is something new, and I'm technologically challenged. So I just figured out how to turn my computer on. So You need to be patient with me. So I'm not sure. I'll try my best to share the PowerPoint presentation. Here it says share, so I'm going to try that. And thanks, Dr. Selvarajan, for the introduction. Again, my apologies for the delay. I didn't know that all the lists that he read, I'm not aware of that. So, thanks for the invitation, Dr. Devappa. I don't think I ever met him, but I appreciate your invitation to speak at this conference. Today, I'm going to talk a little bit about some of the research that is going on in our lab, mainly focusing on plant virus interactions at the molecular level. So we've been doing quite a bit of genomics and transcriptomics, proteomics, and metabolomics work using a couple of different viruses. One is a group called TOSPO viruses and another group called proteoviruses. Those of you who are not very familiar with all these virus groups. So today I'm going to use TOSPO virus as an example and how we are trying to understand the interactions between TOSPO virus and their hosts, in this case tomato, which is again an economically very important crop, and use omics studies to understand not only the interactions, but then use that information to come up with ways to identify resistance genes to these viruses. Basically, omics approach, it turned out in our experience, really fast-tracked the screening for resistance and potentially identifying markers for resistance genes. So that is the biggest advantage of what we are finding with the omics approach. And of course, another important information that we are getting from this omics approach is that we are finding targets both in the virus and also in the host. Potentially, we can edit using CRISPR, because the application of CRISPR fundamentally depends on knowing what targets to edit. Right? So without that, CRISPR is useless. So you can do shotgun using CRISPR, but knowing the target really makes a difference. So some of the benefits or outcomes of omics approach in our experience is that generating information that has applications downstream, such as using CRISPR. So I think I have some background static. I think I was given 30 minutes, but I started late, so I'll try to make up. And I think it must be 10 after for you in the morning. So I'm going to try to share the screen and see if it works.
I'm sorry to disturb, Dr.
So can you see the screen?
Yeah.
Yes.
All right. Yeah, Dr. Selvarajan already kind of read the title, and I briefly gave introduction, so I'll skip that. And very quick overview of the importance of agriculture, where I am located in Washington State, that is northwestern corner of the country. Those who are not familiar with where we are located, so we're very different from Washington, D.C., which is on the East Coast. Agriculture is #1 industry in terms of economic value, employment, revenue it generates, and the tax base. Even though Washington is more known for Microsoft, Boeing, and Amazon. So very diverse agriculture from apples to wheat. So a lot of annual crops, horticulture crops, field crops, perennials, a wide range of food and feed. Alfalfa is a major feed crop, multi-billion-dollar industry. So it's not only just food, but a lot of feed. And floriculture is very huge. And the point is that almost all of these crops, A to Z, diseases are a major issue, production constraint, whether it's fungal, bacterial, or viral diseases, it doesn't matter, because the climate is very favorable for disease development and disease outbreaks. So Washington State, again, as I said, many crops, they are #1 in the country.
Sir,
Apples,
1 minute, sir. Put it on slide mode, sir.
Oh, sorry. Thank you.
Welcome.
And our A quick background about omics in plant virology. Most of you are familiar with the recent advances in high throughput sequencing, also called next generation sequencing or NGS. So now they both are used interchangeably, HTS or NGS, but basically you are cloning and sequencing everything in a particular a sample, be it an insect vector or a plant or what have you, or any plant part. It could be seed, it could be tuber, leaves, or phloem tissue. So, and you sequence, make a cDNA and sequence everything, and then try to use that information for whatever the purpose is. These developments in HTS or NGS and the continued reduction in the cost, it's really enabled a lot of labs to look at plant virus interactions at a level that was not possible before. And NGS and HTS, either one, yeah, it's being applied to understand both on the virus side, which has been going on for a long time, but more importantly, now we are able to do it on the plant side as well. What is happening in the plant at various levels in response to virus infection. For example, in a known resistant variety versus a known susceptible variety, how these plant physiology and the metabolism and genetic mechanisms are operating in response to virus infection. What we see as an outcome, either highly susceptible phenotype or a highly resistant phenotype. What is the basis behind it? So with NGS, now we are able to actually get some insights into that. On the virus side, of course, we know that plants defend through a mechanism called gene silencing or RNA interference. A few years ago, you may recall that the scientist who dissected and explained this RNA mechanism were a Nobel Prize a few years ago. So NGS gives us
Hello, please unmute. Please unmute. The host, hello, please unmute. It's disturbing.
Mute. All right. May I continue?
Sir, you can, sir. I think, please, all of you mute, please. You have to mute.
Me too, I'll be glad to. So on the plant side, so on the virus side, I'm going to give an example how NGS and omics approaches are giving us very useful information in terms of finding targets for manipulation, for virus resistance or virus suppression in this case on the virus side. RNAi leads to basically breakdown of incoming viral RNAs by the host, which the mechanism, the pathways are very well defined and worked out. So we know a lot about it. So NGS actually is enabling us to help us identify what are the small RNAs that are produced as a result of RNA interference. And on the plant side, NGS is now allowing us to determine what transcripts are upregulated or downregulated as a result of virus infection in different genetic backgrounds, right? A resistant population versus a susceptible population. It could be a breeding line, it could be a commercial variety, doesn't matter. So NGS has that, and the associated omics and bioinformatics is now allowing us to look at plant pathogen interactions in the broader sense, with fungus, plant bacterium, or plant virus. Right? So far, more than 20, now I think it's almost 28 host virus combinations. They are studied using this omics approaches, using NGS and then bioinformatics. As I mentioned, from transcriptomics, we are able to fast track identification of genes for virus resistance, as long as you have a differential population of resistant population and a susceptible population. You can determine the transcriptome changes in each of those in response to virus infection. And we are able to actually identify markers for virus resistance. Traditionally, it involves a lot of backcrossing and years of breeding work. But now with the omics approach, it really cut down the time to come up with a marker for resistance or even resistance gene itself. So that is a huge advantage. And then on the virus side, applying these omics approaches, we are able to identify better targets to for more effective RNAi or gene silencing, or as I mentioned, even CRISPR. So we recently published a paper on that using CRISPR in suppressing Gemini virus replication. So as I mentioned, we have been looking at or working on a group of viruses called TOSPO viruses. So I'm going to use that as an example on how we are using the omics approach to study TOSPO virus and host interactions. And to begin with, we looked at the virus side, how the plant is responding in terms of RNAi and the resulting virus-specific small RNA profiles that we first determined to gain a better idea about the resistance response or lack of it in different cultivars of tomato. And then we used RNA-seq approach and then a couple of different approaches to see how these small RNAs are actually interfering in the plant metabolism and what transcripts are affected because of this viral small RNAs and looking at the transcriptomic changes. So because of the time constraint, I will try to cover both, if not only one. I'm sorry about that. So before I get into that, I first want to acknowledge my lab group and collaborators who actually did the work. And I really like them because they do the work and they let me take the credit. I really like them. So Christian Olaya is a PhD student, just graduated last year. Bottom left picture, the one guy wearing a tie and looking like a gentleman. So it was on his when he passed his defense exam. So he's from Colombia, so he's proudly showing the Colombian flag. And then going toward 3' end, Dr. Ramesh, he was a visiting scientist in my lab, came on I believe, a DBT fellowship, and he did a lot of the transcriptome work. And then lady in a lab coat at a flow bench. Ying Zai, also as a postdoctoral fellow in my lab, did her PhD. She did also quite a bit of the bioinformatics and small RNA profiling work. And I have a long-standing collaboration with Dr. Nina Mittar, professor at the University of Queensland, and her group, especially Steve Fletcher and Jonathan Peters, who are very good at bioinformatics, and they did quite a bit of bioinformatics work in analyzing this data because it's terabytes of sequence data. And also I have a long-standing collaboration with Dr. Stefan Winter in Braunschweig and his wonderful group, especially Paulo Margherita, a postdoc in that group. Some of you are not familiar with the virus system. They are strictly intracellular, obligate parasites. When viruses infect plants, they fight back, as I said, by a mechanism called RNA interference. As a result, the incoming virus and any of its replication intermediates gets chopped down into very small pieces. So that plants defend themselves. So it's been very well worked out. And it is through a process where viral RNAs go through a double-stranded RNA phase. And that RNA phase, double-stranded RNA phase, is recognized by the plant defense machinery. And that gets chopped up into very small pieces, what we call viral small RNAs or viral small interfering RNAs. And it turned out that they play a very significant physiological role in regulating host gene expression. So understanding the nature of these viral small RNAs can really give us insights into how they actually interfere in the host metabolism and also modulate host transcriptome. So as I mentioned, the viral RNA gets degraded into small pieces, but an unexpected outcome that was discovered is that these degraded viral small RNAs actually show affinity to some of the host genes and bind to those genes and either downregulate. And as a result, the plant metabolism is affected, plant physiology is disturbed, leading to what we see as a symptom in the plant. So these are the various steps, as you see, that there are possibilities to interfere through various approaches. So I will quickly go over or skip these role of small RNAs as I kind of gave an overview that understanding the nature and the profiles of these virus-specific small RNAs have give us a lot of information in terms of what host genes are targeted by these viral small RNAs. And hopefully I'll have time to give an example. Right? So RNA silencing, those most of you are familiar. As I mentioned, double-strand RNA of the virus gets degraded, is the target. And that's not only in a localized infection in a leaf, for example, but that signal becomes systemic. So that this RNA status or a stage becomes transported, if you will, translocated throughout the plant. So again, I will skip the mechanism behind the generation of viral small RNAs, a lot of graphics. But now we know that plants defend themselves by using RNAi against virus infection. So now virus evolved to overcome the plant defense by coding for a very specific protein called silencing suppressor. Basically, it suppresses the host defense response. And now we know the mechanism, how the silencing suppressors actually inhibit host defense through various biochemical changes in the host, several host proteins. So we have been targeting the silencing suppressor of tascoviruses, try to understand better the nature of the silencing suppressor at the genetic level and also at the proteomic level, and try to identify what are the critical amino acid residues in the suppressor protein that are required for its activity in suppressing host defense. The idea again is to come up with a designer molecule that will block those active amino acid residues, similar to some of the drugs for HIV. They physically block the RNA polymerase of reverse transcriptase of HIV physically, because the structure of the replicase is figured out. And other option is that coming up with the targets of RNA-based targets, specifically targeting those codons that code for those amino acids that are critical for the silencing suppressor activity. So in other words, to give a upper hand to the plant host defense response, otherwise which gets suppressed by the suppressor protein. So it's a continuing arms race. So basically, about omics approach that we have been doing, focusing on small RNAs produced as a result of RNAi, and how they actually interfere or interact and interfere with the host genes. So about tascoviruses, those who are not familiar, maybe I'll quickly go over. It's a name derived from its type species, if you will, tomato spotted wilt virus. And now there are more than 31, maybe 33 different tascoviruses described from all over the world. They are economically very important. They both cause damage in terms of both yield and also the quality, so making the product unmarketable. So tascoviruses are very unique among plant viruses because they are the only plant infecting viruses in a family of animal infecting viruses. For example, hantavirus or fleabovirus. Many of you probably are familiar with the infamous virus Rift Valley fever. But tascoviruses are the only one because of the genome structure and organization. They are grouped with these animal and human infecting Bunia viruses. So these are a worldwide distribution, and they are also very widely present in Indian subcontinent. They have very wide host range. Both agronomic and horticulture crops are susceptible. In India, groundnut bud necrosis has been known for a long time and has a very wide host range, economically very important, followed by iris yellow spot virus in onion. Whereas in the US, it's a tomato spotted wilt virus and iris yellow spot virus are very important tascoviruses on a lot of different crops. Of course, those of us plant pathologists, we love to look at sick plants all day and feel good about it. So I have to show you some sick plants. This is effect of tomato spotted wilt on tobacco. As you can see on the left one, the foreground plant, early infection leads to death. Tobacco, excuse me, being a cash crop, the leaf is actually affected because of the necrosis, and so it's not suitable for curing, for making either cigarettes or what have you. On peanut or groundnut, again, severe yield reduction, early infection. Tomato, severe stunting, total crop loss sometimes. And in case of onion, again, severe losses. Here is an onion seed field in Oregon, just south of us. A seed crop completely destroyed. Typically about an acre of onion seed crop is almost $100,000 value. So it's a very economical, important, and very profitable crop, actually. So quickly, taskforce are spread by thrips, and the uniqueness about these viruses is that they replicate in the insect vector. So that's why you have two very different hosts for these viruses, plants on one side and insects on the other side. So breaking this life cycle continues to be a challenge because the virus has a host in thrips as well, and it replicates in the thrips vector as efficiently as in plants. So there is actually an argument or a hypothesis that these taskforce viruses actually started as insect viruses because they basically developed this kind of a symbiosis relationship and somehow acquired a gene to infect plants and became plant viruses, which makes sense. So again, a very unique genome structure, RNA viruses, 3 pieces of RNA, large, medium, and small, because of their difference in the size. And like any plant virus, large RNA codes for RNA polymerase for replication, and the virus has a lipid envelope, what you call glycoprotein. So there's a gene for that. And then all plant viruses require a movement protein. So here also NSM codes for the movement protein, for cell-to-cell movement. And then, like all plant viruses, task virus also code for something analogous to code protein or a structural protein, what you call it, N. And then, like all plant viruses, task virus also code for a silencing suppressor that I mentioned. This is to overcome the host defense. Here in this case, it's coded by the small RNA. Each of these RNAs, as I just mentioned, code for one or more than one protein. All of them are essential for virus replication, for movement, for encapsulation, and for transmission. So I will skip about the mechanism of the transcription and translation for the lack of time. But again, the important gene products of this virus, code protein, silencing suppressor, glycoprotein that's involved in insect transmission, and the NSM facilitates movement, and of course polymerase for virus replication. So we know quite a bit of the molecular biology and the gene expression strategies of tomato spotted wilt virus, which is basically the same for all other task viruses. So Christian Olaya, who did PhD with me, he was interested in understanding the biogenesis of these viral small RNAs of TSWV in tomato. And he wants to compare the profiles between a resistant variety and a susceptible variety and see if there are any differences in the way that the virus is processed by the host using this RNAi response. So he specifically looked at inoculated leaves very early on in the infection. What is the host response to infection? And we had a very nice system. So we had a very nice system where TSWV resistance is present in many of the commercial cultivars here in tomato. The gene was identified, cloned, and sequenced years ago. It's called SW5. It's a single dominant gene, gives very good resistance to TSWV in tomato. So essentially, resistant varieties, the virus infection just is restricted to only the inoculated leaf. And the virus does not spread to the remaining part of the plant and does not become systemic. Whereas in varieties that do not have this SW5 gene, the virus spreads to the rest of the plant and becomes systemic. So we have a very nice differential cultivars, if you will. One is highly resistant, one is highly susceptible. Right? And because of this one gene, SW5. All right. So again, the graphics here basically shows what I just said. In a resistant variety, the bottom one, virus remains in the inoculated leaf. The top one, a susceptible variety, virus becomes systemic. So then she used omics approach, NGS, followed by bioinformatics analysis to look at what is happening in the inoculated leaf in a resistant variety versus a susceptible variety. So the two varieties that I'm going to talk about in the rest of the talk, the resistant variety, the commercial variety called Drug Defender. So the name kind of self-explanatory. It's very good at defending itself. And the highly susceptible one, where the resistance gene is not there, the variety's name is Marklo. Just FYI. So as I just mentioned, Christian wanted to see if there are any differences in the way that the virus is processed by these different varieties, different genetic backgrounds. Right? So basically, the treatments are very simple. Two varieties, and each one virus inoculated, the other one is buffer inoculated, and then samples are collected at different time points. Of biological replicates and technical replicates. So I won't go into the details. And then after certain time points, leaves were harvested, total RNA extracted, and then enriched for small RNAs, cDNA libraries prepared and sequenced and using the NGS approach or next generation sequencing. So here are some pictures of the symptoms of different, the two varieties. One is highly resistant, one is highly susceptible, just FYI. And most of you are probably familiar, I'm sorry for the clock in the background. Many of you are familiar with the Bioinformatics Pipeline. Once you get this sequence data, you first clean up, remove all the adapters, and do the quality control. And then we use a set of software packages to build contigs and also to remove the host sequences. Tomato genome is sequenced, so we use that to filter out all the tomato sequences so that what we end up with is only TSWV sequences. These are small RNAs. So how small, you may ask if you are not familiar. These are anywhere from 17 nucleotides to 24 nucleotides. That is the typical range. It has been found that is the result of RNAi, the host defense response. 17 to 24, how that's small. So, and based on the contigs, of course, we also try to assemble the whole TSWV genome from these small RNAs. This is a standard bioinformatics pipeline that typically for RNA-seq or omics approaches use. So here are some results. Typically what, as I said, the highest number of cDNA sequences that we got from these samples, the range was between 20 to 25 nucleotides, but the majority were either 22 or 24. And again, it turned out that this depends on the host virus combinations. In some host virus combinations, you will find more of 21 nucleotides. Whereas in this case, it turned out it's 22 and 25, both in resistant and susceptible varieties. Right? So, and then we looked at individual, as I said, these viruses have 3 RNAs, large, medium, and small. So then we separated all these small RNAs, these are millions of sequences, categorized them, which ones are specific to large, medium, and small RNAs. And it turned out that in case of highly resistant variety, Red Defender, the large RNA was targeted specifically, resulting in a highly, significantly high number of small RNAs. Whereas in case of Marglo, which is a susceptible variety, small RNA was targeted. So it made sense once we analyzed all the data that it is consistent with their phenotype, resistance versus susceptible. So basically that's a Christian's goal, compare resistance versus susceptible. Are there going to be any differences in the way that these small RNA profiles turn out? And they did. So the significant difference is that in a resistant variety, a large RNA was preferentially targeted. In a susceptible variety, small RNA was preferentially targeted. Again, bottom right is just a cartoon showing large, medium, and small RNAs. So large RNA codes for RNA polymerase, as I mentioned. The small RNA codes for the silencing suppressor. So and again, what is the percentage of these viral small RNAs for each of the genes? RNA polymerase, glycoprotein, movement protein, silencing suppressor, code protein. Again, there are significant differences in terms of resistant versus susceptible varieties. What is the percentage of these viral small RNAs? Meaning that the host RNAi targeted a particular RNA depending on whether the host has a resistant gene or not. So there's a significant difference in the RNA profiles, small RNA profiles. And again, RNA polymerase consistently was targeted in the resistant variety, whereas that's not the case in a susceptible variety. So again, makes sense. The RNA polymerase is critical for replicating the RNA and making more virus in the infected plant. And in a resistant variety, SW5 gene, so that particular genetic background is targeting specifically RNA polymerase and chopping it up. That's why we have more of viral small RNAs from RNA polymerase gene. As a result, virus replication is suppressed. That's outcome of the phenotype is resistance. Makes sense. So, and more of the analysis confirm the same. That in a resistant variety, you can see hotspots in the viral RNA, in the RNA lRNA that codes for replicase. Whereas in a susceptible variety, the hotspots are the targets in the viral RNA or in the small RNA segment. So consistently, this pattern remained between resistant and susceptible varieties. So information is very useful because now we are able to target or select regions from the lRNA and the small RNA to come up with dsRNA probes for efficient RNAi. And also down the road, our goal is also to use these as targets for CRISPR. So most of this fundamental data or information, it's translational. So we are hoping to use this information to then use it for CRISPR and come up with develop virus resistance. So again, some validation showed that in resistant variety, the relative levels of the viral RNAs are very low compared to susceptible variety, which is expected. So it's just a validation that resistant variety is able to suppress the virus replication very efficiently compared to a susceptible variety, which is expected. So no big deal. And interesting thing is, individual genes he tried to quantify between resistant and susceptible varieties. And it turned out that in a susceptible variety, the N gene is expressed to a very high level, followed by the movement protein, which again makes sense. Because a susceptible variety, the virus becomes systemic. The whole plant gets infected. That means it needs movement protein at relatively high titer to facilitate the movement. And so that's what it actually reflected in terms of the relative levels of these transcripts for these genes in a susceptible variety. And in case of a resistant variety, the NSM movement protein is significantly less. Again, inoculated, the infection remains in inoculated leaf in a resistant variety. That means the host is restricting the movement of the virus from inoculated leaf to rest of the plant. So the relative levels of movement protein here is much less in a resistant variety, which again makes sense. So quickly conclusions, the small RNA profiles of TSWV between resistant and susceptible variety are significantly different. Different parts of the viral genome are targeted in these two different varieties. The relative abundance, the number and percentage of the viral small RNAs, again, significantly different between resistant and susceptible varieties. And the hotspots that the targets in the viral genome again, is the difference between resistant and susceptible varieties. Again, this gave us some insights into what kind of pathways or transcriptome changes or genes are potentially modulated, either upregulated or downregulated in the host. So far, I only talked about the virus side, but the more useful information, if you will, actually is on the plant side. How the plant genes and the various metabolic pathways and the plant metabolism is affected as a result of virus infection. And this is, again, down the road, it's not only to be able to identify resistant genes or markers, but equally importantly, if we can identify a susceptibility gene in a susceptible variety, and that variety happens to be very popular with growers and consumers, now there are possibilities of using CRISPR to edit that susceptibility gene so that you are changing the phenotype into resistance. So that's where real application or usefulness or benefit of this omics data is to identify the susceptibility genes in a desirable cultivar and use genome editing like CRISPR to improve the trait, change it from susceptible variety to resistant variety. So quickly, I will summarize it. I think I have 5 minutes now. I think, or even less. Dr. Ramesh, that I mentioned in the beginning, what he did was really creative. He took all this small RNA data, and then he looked at the tomato transcriptome and used a series of bioinformatics software and in silico approaches basically to see if any of these small RNAs from virus, tomato spotted wilt, have any propensity or affinity to bind to tomato genes or transcripts, and whether those transcripts and the pathways have anything to do with susceptibility resulting in disease phenotype. Right? The idea again is then use those host genes or transcripts, as I just mentioned, can we then manipulate them for trait improvement, but introduce resistance. So quickly, what he found was for each of the TSWV genes, he then looked for transcripts that these viral RNAs potentially can bind. He identified a bunch of them in the tomato genome, the transcripts that are either upregulated or downregulated, and then he identified and listed a whole bunch of various transcripts potentially coding for, as expected, kinases and also cytochrome and several proteases and several transcription factors. Again, most of them are known to be involved in defense response or viral RNAs binding to these transcripts and modulating the gene expression, thereby affecting the host metabolism. Again, there's a lot of data. I don't want to list each and read every one of them, but many of the genes in the tomato genome were actually have the potential of being interfered or being bound by these TSWV small RNAs. So he then went ahead and again, more and more lists of what are the host genes that are modulated, either upregulated or downregulated by TSWV small RNAs. So I won't go into the detail, but then he validated that by selecting the top 10 genes, if you will, or the transcripts that are upregulated or downregulated that he found using in silico analysis. And then using qPCR, he validated that yes, indeed, in TSW infected tomato plants, either resistant or susceptible, these set of genes are either upregulated or downregulated, depending on the genetic background. So again, this gave us some idea about the potential markers, if you will, for resistance, and more importantly, or equally importantly, potential targets that contribute to susceptibility to the virus. Again, so that is again very critical if we want to be able to use CRISPR to target that and modify the trait. So I won't go into details about all the validation data, but conclusions, TSW derived the small RNAs target a wide range of host genes. most of them are involved in metabolic pathways, and many of them are involved in resistance response pathways. And so these small RNAs not only determine the pathogenicity, but also they determine the outcome of the virus-host interaction, either resistance, one outcome, or susceptibility, the other possible outcome. And just like the small RNA profiles, what Ramesh found was these tomato varieties, resistant versus susceptible, they had a very significantly different expression levels of selected target transcripts, depending on the genetic background, resistance versus susceptibility. So, as I mentioned, it turned out like in other virus host systems, TOSPO viruses also through their small RNAs, they do interfere and modulate the host machinery, leading to either disease or in resistant varieties, less disease, if you will. So I will stop here and acknowledge the funding from USDA and NIFA and a whole bunch of other funding sources, and WSU, and also from California, and of course the German foundation, the Humboldt Foundation. And again, I thank Dr. Devappa for the opportunity to talk a little bit about our some of the research projects. And again, I apologize for the delay.
Thank you, Dr. Prabhu, for a wonderful talk on PSWB, where you have used devomic approaches to understand the host virus interaction and looking at the resistant and susceptible varieties, the reason for it in the virus side of virus side, and clearly explaining the RNAi mechanisms wherein the resistance is being explained as the susceptible. Now the presentation is open for discussions. I think we can take up a few questions, one or two questions, please. If anyone wants to ask questions, they can unmute and ask question.
Sir.
Yeah, Shankarappa, please.
Shankarappa, sir, you mentioned that susceptible genes targeting is a new approach. It is a new approach, no problem, sir. Any successful example is there for targeting susceptible gene?
Good question. So I think in case of for potato virus Y, I know a couple of labs are working on that because the potato genome is cloned and sequenced, and so there's quite a bit of known between, and also there's what are the transcription elongation factors in potato that play an important role in virus resistance against potato virus Y. So I think there's some progress made in that in case of potato and PVY. So again, I haven't talked about our work on potato and PVY and our approaches to using CRISPR for PVY resistance, but we're also trying that.
Thank you.
We have several potato cultivars that are highly popular, industry standard. Like russet Burbank, almost 40% of US potatoes is russet Burbank, and it is highly susceptible to potato virus Y, potato mop top, tobacco rattle, but still industry loves it. Companies McDonald's and Burger King, they really like it for the processing qualities and the quality of the French fries and what have you. So the growers still grow it, even though it's highly susceptible to some of these viruses. So yeah, to answer your question, yeah, a lot of labs are actually working on that. Their possibilities are endless. So if there's a partially dominant gene, can you make it more effective in terms of the level of resistance? It's possible. Yeah, that's a good question. Thanks.
Thank you, sir. Thank you, sir.
I have a small query. This is Vipin Hallan from IHPT Palampur.
Oh, hi, Vipin.
Hello, Doctor. How are you? It's a long time interacting.
Right,
nice to hear your talk, and it was a really exhaustive information that you have given. one small kind of query like, are we in a position actually to identify some susceptibility genes which can be utilized for resistance to viruses?
Yeah, good question. Again, it comes down to the differential populations in the phenotype. That's a critical thing, the starting material. So either it's a established cultivar or a breeding material, breeding lines, resistant versus susceptible. And I think it is possible. Again, through marker restricted selection, for example, and traditional back crossing, maybe it is possible looking for those alleles.
Yeah, I think that would be the best way if we can identify some susceptibility genes and then genome edited them and then have the resistant varieties. That would be the ideal kind of situation, I feel.
Exactly, because typically most breeding programs here, they consider disease resistance as an afterthought. Whether it's potato or apple or onion or whatever, most of the features or traits for selection or yield, appearance in case of tomato or pepper, gross weight, and those are the ones. So the breeding lines are advanced in the program now, and then they think of screening for resistance. So as a result, most of the popular varieties in many parts of the world, here in the US, it's a very good example. Most of the popular varieties that industry loves, they tend to be susceptible to one disease or another. It could be fungal, it could be bacterial, or it could be viral disease. Potato is the best example in our case. Most of the popular varieties are susceptible to viruses. But now, and so that's why in this case, all those popular varieties that are susceptible, if we have a high throughput kind of a screening to identify those susceptibility genes, then it's worth trying CRISPR, for example.
Thank you so much.
Thank you. And there is one more question from student side. I would ask that questions on behalf of them. That is, what is the mechanism of symptom expressions? Are there any genes behind the expression of TSWV symptoms?
Good question. Well, traditional wisdom, the result of symptom expression is essentially, in most cases, most viruses, they affect the chloroplast synthesis. And as a result, what we see, the breakdown of chlorophyll as mosaic. A lot of work is done in case of TMV. The TMV actively replicates actually in chloroplast, leading to chloroplast degradation. So that's why we have these dark and light green islands, the classic symptom of mosaic. So the symptom, what we see as symptom could be a result of, again, the host-virus interaction at various levels. If in case of DNA viruses, it's happening in the nucleus. In case of RNA viruses, definitely in the cytoplasm with various organelles. And of course, these virus-specific small RNAs, now we know that they play a very important role in binding to selected host genes and either shutting their expression or downregulating. So what the result is, all these kind of symptoms, either stunting of the plant or wilting or mosaic or what have you.
Yeah. Thank you, Dr. Prabhu, and thank you for a wonderful talk on this. And this is around 1,000 students are participating in this particular webinar, and your talk would have been the talk is very, very useful for the students who were listening to this, though they could not interact too much here. And thank you very much for sharing a wonderful experience what you had with the TSW and with your team. Thank you very much. Let us go for a next speaker. The next speaker is Dr. Narendra Kadu. Dr. Narendra Kadu, are you here?
Yes. I'm here,
Yeah. So since we are running short of time, so I straight away request you to go ahead your presentation, and you will be talking on omics. Dr. Narendra Kadu from National Chemical Laboratory would be talking on omics analysis of chickpea fusarium interaction, reveals differential modulation of plant defense and fungal virulence strategies. Dr. Narendra Kadu, you can go ahead with your presentation. We will have time for discussions later. Thank you.
Okay, okay, correct. So I'm now sharing my screen. Is the presentation visible?
Yeah, very much.
Okay, yeah.
Very much. Go ahead, please.
Yeah. Okay, so I thank the organizers for inviting me for this as a keynote speaker, and I'm pleased to be here. And I am actually very much happy to give the seminar. So I'll try to complete in about 25 minutes because I know that we are running short of the time. So I'll be talking about the omics analysis of this chickpea fusarium interactions, so which reveals the differential modulation of plant defense and fungal virulence strategies. So just a briefly introduction, like the chickpea we know is a kind of a self-pollinated plant, which is a diploid, and genome size is about 740 MB, and it is a third most widely grown food legume, and it is most important pulse crop of the Indian subcontinent. And it is rich in protein, so it is called as a poor man's meat. It is being a legume, it increases the soil fertility by atmospheric nitrogen fixation, whereas however the average chickpea productivity is much lower, so which is because of its like susceptibility to various biotic and abiotic stresses. So we work on this fusarium build, and we have been working since almost like past 2 decades. So initially we worked on the molecular markers, but later on we moved to the omics approaches. So you can see that in the left hand side of this field, you can see that the wilt resistant genotypes are there, and this photo has been taken in about 2 weeks after sowing. So you can see on the left hand side there are these wilt resistant genotypes which are which are growing nicely. Whereas on the right side of the field, you can see that many of the plants have, or most of the plants have died. Some plants are there. Now this is like 2 weeks within photographs of 2 weeks, but this wilt disease within 3 weeks, within say about 21 days, it kills the susceptible varieties every time. So that means there is a which results in damage of nearly 90% within 3 weeks. So suppose the farmer has grown the variety and which is susceptible to the wilt, then he will lose almost like entire field he will lose within 3 weeks. So this pathogen causing the disease is called Fusarium oxysporum, Hormus species Siceri. So in short, it has been named as, like I'll be referring to it as a ecclusive hawk. So now this pathogen is characterized into 8 races, and of which in India there are 4 races are present. So 1A, 2, 2 and 4. 3 races are present, and 1A is the most prevalent in India. And this pathogen, it infects chickpea through roots. So as I said, up to 90% of the disease, 90% of the losses could be there due to in the disease favorable condition. So how do we control it? So mostly we can control it using the like chemical fungicides, which are actually toxic, and they also lead to emergence of resistance into the pathogen, and they're expensive. However, in case of chickpea, I mean particularly this wilt disease, this disease is a soil borne. So that means we cannot spray the fertilizer, we cannot spray the fungicide, but we need to do the soil fumigation, which is very, very expensive. Then there are some biocontrol strategies, such as some of the like fungal flora, like some of the microbes, they can be used for controlling the pathogen, Fusarium oxysporum Siceri, which is pathogenic. So we can use non-pathogenic fusarium species as well as Trichoderma, Pseudomonas, etc. But then the most work has been done in case of these using the classical breeding approaches. So which is to breed the disease resistant variety. The classical breeding takes a lot of time. So we can use the molecular breeding strategies. So which means we need to identify the disease resistant genes, and then we use the genes for breeding for disease resistance. However, this is quite, I mean, not that easy that said. But anyway, we use both these approaches. We can develop the wilt resistant cultivars, which are more sustainable. Then the coming back to the wilt. So this fusarium actually enters into the xylem vessel. So xylem is the battleground for the plant and the pathogen. So this hawk, that is pathogen, it proliferates in roots and it blocks the xylem vessels. So you can see here, and then so because of when the xylem vessels are blocked, it hinders the water uptake by the plant, and then that results in the wilting as well as final death. So if you see, in case of the susceptible varieties, in case of susceptible variety, this is yellow colored, like this in the cartoon, yellow colored is fungal hyphae. So they can go to the roots to the shoot, and then they can block the xylem vessels, and then which leads to the wilting. Whereas in the resistant varieties, so the fungal hyphae, after they may be able to enter the plant through the roots, but then the plant can successfully produce the proteins like accumulation. There is accumulation of the proteins such as callose as well as stylose, and then many other proteins are there, these resistant proteins. So they actually prevent the pathogen hyphae from growing up, and then they restrict the pathogen growth, and then the plant is able to almost grow almost normally. So to study it further, we did a greenhouse experiment. So where we use two varieties. One is called JJ62, which is wind susceptible, and then there is another variety that was developed in this program only, which is called Digvijay. So these varieties were grown. Then we had a control experiment, like a control treatment, and then inoculated. So these JG62 plants were inoculated, and then this is designated as JGCs, JG62 control, and JG inoculated is called as JGI, and likewise it is DVC and DVI. So after the inoculations, we grew them about a few days, and then we collected the tissue at different time points. So time points are 2 days after inoculation, 4 days after inoculation, 8 days after inoculation, 12 days after inoculation. So in all cases we had, and plus we also had a 0 day, that is a initial stage that we also had. So these are the some of the photos of the plants which are taken at different time points. So we have collected both shoot as well as root independent. So we have cut the root as well as shoot after harvesting. So this photos show the photograph shows after the various time points. So you can see that the right now 2 days after as well as 12 days after the harvestation. And as I was talking about in that cartoon regarding this deposition, so you can see that in case of the resistant variety, so in the control there is no pathogen, either in the DVC or a JGC there is no pathogen. So there is no like the fungal, there is no kind of a deposition of the callose or lignin deposition. Whereas in the resistant inoculated plant, so you can see that there is a progressive increase in the deposition, and then the vessels are becoming smaller and smaller. But still, they are still open even after 12 days. But in case of susceptible variety, the vessels are completely blocked within 12 days, and then there is a complete blockage, and because of which the plants are wilt and they die. So to study the further, we perform we can use different approaches such as genomics approaches, transcriptomics approaches, proteomics, as well as metabolomics approaches. So all of these omics approaches can be used to study the plant pathogen interactions, and they can reveal the different we can say pathogens, different genes or proteins or metabolites which are involved in the. So we perform the excuse me, can I request everyone to mute their microphones? Please mute your microphones. Yeah. So then we perform the to study to visualize the actual disease progression. So we perform the confocal microscopy of the inoculations, and for that we developed kind of a construct using eGFP construct, that is enhanced GFP, and then that was transformed into agrobacterial, and then we use agrobacterial mediated transformation to transform the fusarium of spores, and then this pathogen, which is eGFP pathogen, which was used for pathogen inoculation. And then this picture shows how the pathogen looks like. So this is the A panel shows only the pathogen in culture. So it can show it shows a very good green fluorescence, and then we inoculated. So B as well as other panels show the pathogens which is in the root. So you can see the red color is actually with the plant, and then whereas the green, or you can see sometimes the yellow color, so that is the pathogen. So you can see that how the pathogen is growing in different tissues. And then this is a temporal expression pattern. So the first, like the A panel is shows that 1 to 3 dpi plus 6. So you can see that how the pathogen is growing, and then slowly it is going on inoculating. So this is at 10 and 12 days after the inoculation, the pathogen is growing. Then at again if you see by the 25 days after the inoculation, in case of JGI, you can see that there is almost like the entire blockage, the complete blockage is there of these xylem cells. Whereas in case of this DVI, we can see the pathogen only in some few cells. Okay, so pathogen is still there, but it has been severely restricted by the plant, which is a resistant plant. Okay. So now these are the roots, and on the right hand side you can see the shoots. So in case of this VGA, again there's a pathogen has actually gone into the shoots, and even in the upper shoots there is a complete blockage. Whereas in the plant, resistant plant, there is a very small spores here and there you can see, but otherwise it has been the pathogen has been mostly restricted. So if you see the late stages, so again it's the same phenomena. You can see that the pathogen everywhere in case of this susceptible variety, whereas in the resistant you can see the pathogen barely. You will have to search for the pathogen. So likewise, there is so much of pathogen has been restricted by the plant. Then this is visually we could see, but then we wanted to see quantitatively. So for that, we perform the qPCR analysis of the fungal colonization, and then we have performed the real-time PCR. But this real-time PCR is with DNA, pathogen DNA. It is not pathogen RNA, but pathogen DNA. So we can quantitate the amount of pathogen in the plant. So you can see that say after 7 days, there is so much of increase in the pathogen load in case of the susceptible inoculated, whereas in case of resistant lines, which are blue bars, so it is a pathogen even very much low is there. So almost like right from the beginning till the end, there is hardly any pathogen which can be seen in the resistant plant. Then we also perform the analysis of the gene expression analysis. Now these are the expressions of the fusarium gene. These are virulent genes from the pathogen. We can see that the genes such as chitin synthase, G protein subunit, mitochondrial carrier protein, as well as many other proteins, many other genes, they are highly upregulated in the susceptible variety. So, whereas in the resistant line, there are only few genes are present, few genes are overexpressed. Then we again wanted to perform the detailed analysis. So we use the transcriptomics approach using the SAGE technique. It is called serial analysis of gene expression. So this is actually, this is a little older technique. So now it has really become outdated, but then still we use that technique. So because when we started this experiment, so the RNA-seq was not that prevalent. So we use this SAGE technique, and it is little complicated techniques. I'm not going into the details. So I'm just coming directly to the analysis for the short of time. So about a large number of genes were identified which were differentially regulated between the resistant inoculated as well as the susceptible inoculated. And then the genes were clustered into 6 different clusters. So the cluster 1 was upregulated in the DV, that is resistant plants. The cluster 2 was upregulated in the resistant, and it was downregulated in the susceptible, whereas cluster 2B was downregulated in this resistant inoculated and upregulated. So likewise, different genes were identified, differentially expressed genes were identified, and then those genes were analyzed data. So when we analyze all that data, so a picture which is a kind of a quite complex picture emerged. And then there were a few kind of pointers such as the genes involved in photosynthesis, redox reaction, or abiotic space were upregulated in the resistant parent, whereas other genes such as those involved in the some post-translational mechanism, as well as some other genes which are involved in translation, ribosomal structure, biogenesis, were also upregulated. So again, like this data looks quite complex. I'm just summarizing that. So we found that about 562 chickpea genes were expressed only in case of this JGC52, which is pathogen, which is susceptible. Whereas in case of the resistant line, there were about 860 genes were upregulated, and in case of the susceptible plants, the genes which are actually the genes which are upregulated, so many of the genes were downregulated, whereas in case of susceptible, in case of the resistant lines, so most of the genes were upregulated, which are involved in disease resistance. So then we have also performed the QRT PCR validation. So real-time PCR was performed for all the genes, and then so that's why we have re-validated the expression of these genes in the resistant, susceptible, as well as control varieties. So then we also analyze the pathogen transcriptome. So the previous slides were for the chickpea transcriptome. So now this is pathogen transcriptome. So interestingly, we found that about 533 genes were expressed only in the susceptible variety, whereas in the resistant variety, only 5 fusarium genes were expressed. So you could see that there is nearly 100-fold change. So in case of susceptible, more than 523 genes were expressed. So again, likewise, some of the genes which are listed here, which are present in, these are 18 genes which are present, which were expressed in both susceptible as well as resistant plants. Then we also perform the different statistical analysis. So these are the standard graphs that you get using a blast or any of the kind of geo annotations. So I'm just showing one as a kind of a representative, but we performed a lot of different analysis, and then the picture which emerged was quite complex, and there were a large number of genes which are these are actually involved in the cell wall degrading enzymes or the transporters, or the genes involved in photosynthesis, carbohydrate metabolism, signaling. A large number of genes were going to be expressed. So the main problem with this omics data is that you get lots of data, and then you need to really work. You really need to identify which are the most significant genes or the key genes, and then those you need to focus, and then you perform the functional validation. So that is already stated, and then so these are the long stage summary using the long stage. So where we found that the definition is chickpea transcript as well as pathogen transcripts were identified. Genes were overexpressed in the susceptible cultivar only, and then the expression analysis of the plant depends genes that were found to be expressed only in the resistant variety. So this is published in scientific reports, and then we perform the metabolomics analysis. So metabolomics was performed using two approaches. One is NMR based, and later on it was LCMS based. So the case of NMR metabolomics, so this is a workflow where we take the tissue and we perform we generate the NMR spectra, then that is analyzed. The statistical analysis is performed. Then after the statistical analysis, you get the different metabolites which are going to different kinds of can say different graphs you can get, and then you can identify the metabolites which are involved either in resistant or susceptible. So again, I'm skipping all the procedure or the other data analysis that we have performed and directly coming to the what matters, the results. So you can see that the left chart, which is resistant variety. Excuse me. Yeah. So the resistant variety, in case of resistant variety, you can this graph they show what are the metabolites. These are in fact each spot actually contains the thousands of metabolites. So each or so that means at 0 day time point, so all the metabolites they were grouped here, whereas at 2 days after inoculation, 4 days after inoculation, 8 days after inoculation. So if you see these short, small kind of this graphs here, so that indicates that you can just see how the trajectory of the metabolites is happening. So right from this 0 day to 12 days. So this is a trajectory in case of resistant cultivar. So this is a control, red one, and green one is inoculated. So both of them are almost like following the similar strategy. But in case of the susceptible variety, you can see the trajectory is completely reversed. So in case of this control, you can see the control strategy control will be same, whereas the inoculated case, so this trajectory of the metabolites is completely reversed. So they are going into opposite direction. So that means here only it says these small graphs only indicate that there is something drastically happening in the susceptible variety. Then these are some loading plots or score plots. So again, rather than explaining the complexity, I'm just explaining the score plots. You can see that the metabolites they are grouped into two different distinctly they are grouped into two different classes. Now this is from the Digvijay control. This is Digvijay inoculated. So you can see that there is a clear-cut differentiation between the metabolites. Again, here also each spot can contain about hundreds or thousands of metabolites. Now the same thing we found in case of this inoculated case also, that is JJ62. So there is a clear separation of the metabolites. And so what do we do with all this data? So it's a large amount of data, but then what do we do? So finally, then we need to derive the meaning from that, and this chart indicates you what is actually happening. So if you see that there is in case of the susceptible variety, there are big red patches, so that indicate that there is a decrease in expression. Indicates the decrease in expression of the metabolites. So you can see that particularly if you see the amino acids and sugars, they are decreased in case of the susceptible variety, and there is sometimes no change or there are some upregulation in the resistant variety. Whereas the flavonoids, which are known to be involved in the disease resistance, so they are highly expressed in the resistant case, but they are not expressed or they are unchanged in case of the susceptible variety. So after this, when we do that, so we identified one compound called clotrimazole, which is a antifungal compound, and that was found to be present only in the resistant variety, but it is downregulated in the susceptible variety. So just to summarize this part of the work, so we identified a total of 52 significant metabolites from the complex mixture, and the pathogen inoculation, they cause major change in the levels of amino acid, sugar, isoflavonoids, as well as antifungal compounds. And we presume that the amino acids and sugars, they might be involved or might be utilized by the fungus for their survival inside the host. And then we also identify the antifungal compound called clotrimazole, which is found only in the resistant. Then we moved on to the non-targeted UPLC-based mass spectrometry. And again, you can see that there are about 6,500 metabolites which are identified, up to 6,500 metabolites. And it is LCMSA performed in two ways: electrospray ionization positive mode as well as negative mode. So in each case, we get thousands of metabolites, and these are the different. This Venn diagram shows how the metabolites are the number of metabolites which are found in case of different tissues, such as in case of this Digvijay at 2 time points, 2 days after the inoculation, 4 days, 8 days, 12 days, etc. So again, to make sense, so I'm skipping all the kind of methods and materials. I'm just coming to the results. So you can see that again, like we identified the flavonoids or isoflavonoids as well as saponins. So all these compounds, they played role in the resistant variety. So based on this, because we get lots of data, so we also perform the like pathway mapping, and then we could now these charts here, you can see small charts. They show the gene the accumulation of the metabolites at different time points. So first set shows at 2 days, 4 days, 8 days, and 12 days after inoculation, and then the red one is susceptible, and then the blue ones are the resistant. So you can see that a large number of metabolites we could get, and we could reconstruct the pathway based on the metabolite analysis, and we could see like how the plants or how the resistance really operates in the plant. So to summarize that, so fungal inoculation led to the increase in the intermediates of this metacarpine and meconium synthesis, and we also found that the chitin-induced anthraquinones, they showed higher accumulation in the resistant culture. Then after that, we performed the proteomics analysis, So again, after performing proteomics, we got kind of a large number of genes. So they were grouped into different clusters. And you can see that there are about 8 clusters were identified, and in each cluster there are the parenthesis shows the number of proteins that those are composed of those many clusters. So after these protein and proteomics data were also used to reconstruct the pathways, and there is a legend here. So the upper, you can see the small charts, small heat maps you can see here. In each case, so the legend is here. So first row is for the registrant, and last row is for the susceptible varieties. You can see that there is a differential accumulation of these proteins in case of these different pathways such as glycolysis, the green pathway, etc. Even TCA cycle. So to summarize this, so the Fock inoculation led to a rapid lignification in the registrant cultivar, and then the proteins from the isoflavonoid pathway they played a critical role in chickpea defense. And then we also identified the elevated levels of the proteins from this UPR pathway, and which might be crucial for the plant survival. And some of the resistant proteins, low resistant proteins, such as protein 3 were also detected. So coming to the overall summary, so this we identify the differential colonization of the Fock, the susceptible as well as resistant cultivar using this multiple microscopy, and we found that the most of the pathogen virulence gene they are overexpressed in the susceptible cultivar, whereas in case of resistant cultivar, so there was expression of the protein metabolism which contributed to chickpea defense. So sometimes like the other genes which are involved in signaling as well as hormone metabolism, so which also played a role in chickpea defense. So then we also found that there is a complete cesarean metabolism in the susceptible cultivar, and we found that the pathogen could successfully complete its life cycle only in the susceptible variety, whereas the resistant variety prevented its proliferation and reproduction. So this is the underlying statement that the pathogen could happily grow in the susceptible plant and its complete life cycle, whereas in the resistant it couldn't do that. So future directions are we are now working on this RNA-seq analysis, the serum interaction, so which will give us the genes, and then we also plan to use these genes for functional validation, and then we also want to explore the pathogen transcription factors, and then we also would like to identify the downstream genes as well as some of the pathways which are differentially motivated in the resistant susceptible. And we have also performed the identified the role of microRNAs in the plant pathogen infection. So this we have already done, and we also published it. And so in short, like we would like to perform a combination of metabolomic as well as proteomic and transcriptomic data for the wholesome understanding of the plant pathogen infection. So these are some of our publications. But then, as I explained, like many things they happen at the same time in real life. So real life is complex. So likewise, in each cell also there is a same thing goes on. And you can see that there are extremely large number of genes and pathways, metabolites, so transcription factors, microRNAs, they are involved. So all of these actually contribute to the resistance or susceptibility depending on the which genes are or which proteins are expressed or what is the environment. So many things are different. So it is although it is challenging to make sense of this, but still if you use the right pathway, right tools, you should be able to identify what actually happens in the plant. So I would like to acknowledge our PhD students, and our collaborators, as well as I'm happy to take any questions. Thank you.
Thank you, Dr. Naresh. Okay.
Yes, yeah.
Mute.
Hello, there is a lot of disturbance.
Yeah, a lot of disturbance.
Hello?
Okay, now it's working fine.
Yes.
Okay, it's a very, very wonderful talk, Dr. Narendra Kadu, and it's so good that you have shared the entire work on this Fissarian chickpea model, wherein you have used the transcriptomics, metabolomics, proteomics, and all together to understand the interaction between the pathogen and the host. And the build is the major hurdle in chickpea and banana. Also, it's a very big problem. Now, recently, tropical race 4 in banana also, it's making a big havoc in the banana industry. And the work which you have shared is very, very useful to all those students who attended this. And now we can take one or two questions because it's very interesting talk and wonderful slides he showed, and with a lot of data, almost like a decade of research he has shared here. Can one or two questions we can take up? Anyone, please? Any questions? If anyone wants to ask questions, just unmute and ask questions.
Dr. Narendra, good morning. I am Nakiran from TNAU Coimbatore. So the entire presentation was right from the basic aspect till to the latest molecular aspect. So I'd like to appreciate on that particular issue of your very good presentation. And regarding the virulence genes that has been delivered in your presentation, is there any work that has been carried out on 6 genes?
Sorry?
Any work that has been carried out on 6 genes related to FOC in your chickpea?
Yes. Actually, we are taking on those genes ahead, and we are actually trying to perform the functional validation. So we had initiated the work, but then because of the lockdown, that work has been stopped. So we need to reinitiate that. So so it is we are going ahead with that.
Thank you.
Okay, thank you.
Question.
Yeah, please.
Actually, conventional breeding with handling 1 or 2 genes, transferring to desired background, a lot of problems most of the breeders are facing in getting a desired genotype. So much information is generated through the recent technologies and approaches. How to really design or come out with a desired plant type with all the whatever your resistant genotype is expressing the and other molecules. How to really come out with a variety or a genotype with all the desired genes expressed in that background.
Okay, I don't have direct answer, but I will still tell about the little bit about what work we have already done. So as I say that we have been working since last almost 2 decades. So we worked with this Mahatma Phule Krishi Vidyapit Prabhu. So they did all the field work and all. So initial work what we did in our laboratory was on molecular markers. So we identified several molecular markers. And then those molecular markers which are tightly linked to the resistant genes, so those were handed over to the Mahatma Phule Krishi Vidyapit Rahuri, and then as well as they are all available in the public domain. So that MPKV Rahuri, they have used those genes for marker state breeding to develop the new varieties. So likewise, other even in this North India also some scientists they have used the markers and they have developed the varieties. So that was using molecular markers. However, this using the genes it will be good. So, but then at the moment we cannot use the genes so we cannot use the genes for transgenes we cannot be used. So the other approach would be like when we identify the genes that genes which are involved in resistance. So we need to develop the markers for that genes which are gene specific or genic markers. And using these markers, we can do the pyramiding of the genes into varieties. So that way, we will be able to combine the various resistant genes into a single variety. So this is non-transgenic and purely breeding approach that can be used. But I just want to add is that NCL I mean, we don't have, we have good lab facilities, but we don't have the field facilities. So we collaborate with the agriculture universities, and then most of the work happens in that. So we have been highly successful, and if somebody would like to collaborate, we'll be happy to.
Thank you, Dr. Narendra Kadu. It's wonderful, and I am happy to know that you are from IRA. You are also now the IRA alumni.
Yes.
You were genetics and plant breeding at both masters and PhD there, and very good experience. Thank you to hear your wonderful talk. Now I would go for You can just you can stop your sharing this one.
Yes. Stop.
Yeah, please. Yeah, thank you.
Okay.
Now I think we have got the 5 oral presentations. So I request Raghavendra to give a talk on soil. Some echo is happening. What is that? Something is. Hello. Yeah, some echo is happening. Okay, now it's all right. Can I request Raghavendra to give you a talk? Mr. Raghavendra.
Sangeet, 1 minute, sir. Sangeeta, madam.
Yes.
Hello.
Raghavendra, are you ready?
Yes, he is there.
Sir,
It's starting now.
Yeah, tell me.
He's
Sir,
having
maybe I'll
some
present
technical problem. You will present next.
Okay, then we will invite Dr. Pramesh.
Yes, sir.
I invite Dr. Pramesh to give his talk.
Yes, sir.
Pramesh,
Yes,
can
sir,
you
I'm ready,
get
sir.
over? Yeah, please.
Yes, sir. I'm ready, sir.
David now. Yeah, please.
So I'm sharing my video. My slides I'm sharing. Is it visible, sir?
Yeah.
Okay, fine.
Yeah, Nana.
Respected chairman and distinguished scientists from different parts of the world, a very good morning to you all. Now I will discuss a few of significant outcome of research on morphomolecular characterization, comparative genomics, and mating type locus analysis of Pustulogenaria virens isolates from southern India. As we know that rice is one of the most important food crop of the world, but under field condition it is suffered with many fungal and bacterial diseases. In recent time, many minor diseases such as false mud, stem rot, and sheath rot are emerging into an epidemic form. Among them, false mud has caused many epidemics in the country already, and it was first reported from Tamil Nadu in the 1870s. In India, it was earlier regarded as a minor disease, but it assumed already as an epidemic form and threatening the rice production. It causes yield loss up to 11%. In addition to the yield loss, it is also produces some of the hazardous toxins into the grains. It is posing several health hazards to human as well as animals. So, as this disease was regarded as a minor disease, the previous studies are mostly focused on pathogen biology, symptomatology, and yield loss studies. Whereas in 2014, the first sequence of this pathogen was published from China and later from Japan in 2016. However, although the disease and the pathogen was first reported from India, the information on its sexuality, pathogenomics, and virulence structures of the pathogen population is not available. Therefore, the study was undertaken to generate the information on population structure, genetic diversity, sexuality, and also to generate the complete genome sequence resources for the Indian strain. These informations are very essential, especially from the country where pathogen was first originated. So, to achieve our objectives, we conducted an extensive survey in the southern part of the country, and we estimated the potential yield loss in different ecosystems as well as in different cultivars. We from the different ecosystem, we collected more than 100 samples, and from them we isolated 61 distinct isolates. We studied the genetic diversity, mating type, as well as virulence profile. Among the different virulent strains, we selected one of the virulent strain for genomic characterization. This is the passport data of different strains collected from the different geographical locations of the country, and one of the column indicates the disease severity from the field where the isolate was collected. These are some of the pictures showing the symptoms of this pathogen, starting from white, smut ball to up to the matured, the black, dark green, black colored smut balls. These are the